During development, the embryo, it has primordial germ cells. Now these primordial germ cells, they are going to migrate from the endoderm of the yolk sac to the gonadal ridge, which is the developing ovary. So they're ultimately going to form gametes, which in this case is the mature oocyte or the ovum. The process by which the germ cell forms a gamete, that is gametogenesis, and that process is by meiosis. Here it's from the oogonium to the mature oocyte or the ovum, and the process is called oogenesis. To understand the steps of oogenesis, we'll need to know meiosis first, so we're going to go back to a bit of cell biology. There's mitosis and there's meiosis. Mitosis is how somatic cells divide. Somatic cells are diploid cells. They have 46 chromosomes in 23 pairs. That's 2n. So 2n means diploid. And they divide into 2 diploid cells. Now the germ cell, like somatic cells, it's diploid as well. It has 46 chromosomes in 23 pairs. This is 2n, diploid. By meiosis, that number is going to come down to half. That's n, or haploid. This is versus mitosis, where the number didn't change. In mitosis, one diploid cell, it divided into 2 diploid cells. See, there's no change in the chromosome number. In meiosis, after 2 series of divisions, one diploid cell divides into 4 haploid cells, so its number reduces by half. If this germ cell were to form more germ cells, that would be by mitosis. But for the germ cell to form gametes, that is by meiosis. First, let's go over the stages of mitosis. So mitosis has prophase, prometaphase, metaphase, anaphase, and telophase. And in between mitotic divisions, there is an interphase that has a G1, S, and a G2 phase in it. DNA replication happens in the S phase, where the chromosomes duplicate. During prophase, they condense. In prometaphase, the spindles, they destroy the nuclear membrane and attach to the centromeres of the now condensed and visible chromatids. During metaphase, they line up along the equatorial plate. They split at the centromere and move towards opposite poles during anaphase, and they reach the opposite poles during telophase. The nuclear membrane reforms, cytoplasm division by cytokinesis gets completed, and there's formation of 2 cells with the same number of chromosomes as the parent cell. Meiosis is the same, but it's very different. Now, putting down 46 chromosomes is difficult, so we're going to use 4 just to make it simpler to understand. Unlike mitosis, meiosis has 2 divisions, a first and a second meiotic division. Before the first meiotic division, again, the cell does go through an interphase just like mitosis. The DNA replicates during this phase, so there's double the number of chromosomes, which are now called sister chromatids, held together at the centromere. Once the cell enters meiosis, it has the same stages as mitosis with prophase, metaphase, anaphase, and telophase. Now we're going to put 1 next to each phase because this is the first meiotic division. So we start off with prophase 1, and now is when it starts to get different. Prophase 1 in meiosis is longer, and it's got substages: leptotene, zygotene, pachytene, diplotene, and diakinesis. Leptotene is like the prophase of mitosis, where the chromatids, they start to condense, but they're still not clearly visible. So they go from being fine and thread-like to becoming shorter and thicker. During zygotene, homologous chromosomes, they find each other and they pair up. Now this does not happen in mitosis. So they actually lie parallel to each other with a synaptonemal complex in between, but we're going to put them side by side so that we can understand the rest of the steps. This process of pairing, it's called synapsis, and the 2 pairs together will now have 4 chromatids, so it's called a tetrad. Now we move into pachytene, where chromatids from different pairs, they overlap at different points. So this is how exchange of genetic material happens, and the process is called crossing over. During diplotene, that synaptonemal complex, it disintegrates, and the points of overlap, they become adherent, and those are called chiasmata. During the last stage, that's diakinesis, they separate. So now you can see that there has been an exchange of genetic material that happened during crossing over. Now these chromatids, they completely condense, the nuclear membrane disappears by the spindles that are forming from the centrioles. So this phase would be similar to pro-metaphase of mitosis. And now we're done with prophase 1. There was chromatid condensation and crossing over. Now these chromatids are going to line up along the equatorial plate. Sound familiar? That's because this is metaphase. During anaphase, in mitosis, remember there was a split at the centromere? That doesn't happen here. So randomly, one set of chromatids is going to move to either pole of the cell. During telophase, they reach the poles. Then the spindles disappear, the nuclear membrane comes back. Cytokinesis has divided the cytoplasm. So now we have two cells which have half the number of chromosomes as the parent cell. So the first meiotic division is a reduction division. Now we're going to go into the second meiotic division, which is easier than the first because the steps are like mitosis. Again, there's an interphase before this, but there's no DNA replication that happens because they are already doubled. So we'll pick up one of these cells. Now in prophase, the chromatids will condense, and in late prophase, spindles forming from the centrioles, they disintegrate the nuclear membrane and attach to the centromere of the chromatids. They will line up along the equatorial plate during metaphase. They will split at the centromere and move towards the opposite poles during anaphase. They will reach the poles, the spindles will disappear, the nuclear membrane will reform during telophase. And with cytokinesis, the cells have now divided into two. The chromosomes, they uncoil and they become thread-like again. So all of this was in telophase. So one cell divided into two cells, and each of those two cells divided into two cells again. From one diploid cell, there are 4 haploid cells. Now, why did they become haploid? Because after fertilization, that number will go back up to being diploid again. The diploid cell was a germ cell. The haploid cell is a gamete. And now we're going to apply this to oogenesis. The primordial germ cell, it divides multiple times as it migrates, forming more germ cells. So what kind of division is that? That is mitosis. It's not forming gametes, it's just forming more germ cells, so it's mitotic divisions. They migrate to the gonadal ridge, which is the developing ovary. The ovary has an outer germinal epithelium which has cuboidal cells. Below that is dense connective tissue called the tunica albuginea. Then there's the ovarian cortex, which is continuous with the medulla. These cells, they're going to migrate into the cortex, and then they differentiate to form oogonia or the primordial ova. These are diploid cells. The oogonia undergo mitosis to form more oogonia until around 5 months of gestation. After that, some of them can degenerate, but what remain, they undergo the first meiotic division. These are the primary oocytes, but they get arrested in prophase 1 of meiosis during the diplotin stage. So the primary oocyte gets stuck in prophase of the first meiotic division. This is meiotic arrest. This is the situation at birth. So everything till now was until birth, and they stay in this stage until puberty. At puberty, just before ovulation, there's a surge of the luteinizing hormone. Under that influence, one primary oocyte completes the first meiotic division, and it forms a secondary oocyte, which gets most of the cytoplasm. The other cell is called a polar body. It's the first polar body. Now, each of these have half the number of chromosomes because remember, the first meiotic division is a reduction division. The secondary oocyte, it quickly enters the second meiotic division where it gets stuck again in metaphase. So this is the second meiotic arrest, and it gets stuck here until fertilization. Only if fertilization happens can the secondary oocyte continue the second meiotic division and become a larger mature oocyte or an ovum, which again gets most of the cytoplasm, and the other cell is a second polar body. The first polar body, that could either degenerate or it could also go through a second meiotic division to form 2 more polar bodies. But ultimately, all 3 polar bodies are going to degenerate. And what remains is just one functional mature oocyte in the end, which is the gamete. And this has half the number of chromosomes than the germ cell that started the process. So all of this, that was oogenesis. And the oocyte, that is inside cells. So there's a coat of cells around it forming a follicle. And the follicle has different developmental stages. All of that is folliculogenesis. And together, under the influence of hormones, they form an ovarian cycle. So that is meiosis and oogenesis. If this video helped you,